All right, welcome back, guys. Just going to go ahead and get started here a little bit. Again, if you haven't seen a live one before, lecture, you can post in the chat, and I will be able to see it because I have two monitors going now. So if you have any questions, just feel free to type them in as we go. We're going to wait just a few minutes here to get everybody that wants to be here online. If you have any questions from last time, go ahead and post them in the chat while we're waiting. Or if you just want to say hi, go ahead. All right, well, we're going to go ahead and get started. It's 1 o'clock, so I won't waste any of your time. So here we go. This time we're going to talk about translation. So we talked about transcription. Again, when we talked about transcription, we're going from DNA to messenger RNA. So basically taking the DNA code and turning it into a RNA code. And that RNA code is a lot shorter than the DNA. DNA can be several hundreds of thousand nucleotides long. RNA, we're talking maybe 100 to maybe 1,000. If it's a really big gene, maybe 10,000 nucleotides long. But again, we're going to take that code and we're going to decipher it into a polypeptide chain, and that's going to fold and make our protein. So the question is, how do you go from a messenger RNA to a polypeptide chain? Again, we are going to use codons. Codons are 3 nucleotides long on the messenger RNA, and those codons code for a very specific amino acid. All right. So as we look here in the photo, we will see that there are 3 nucleotides, AUG, and that represents a specific codon. So that adenine, uracil, and guanine all code for a specific codon, which in this case is methionine. Then the next 3 are red, and that codes for another amino acid. And we continue to do that and link these amino acids, which are molecules together in a sequence, and they create this long chain. And if you forgot what amino acid is or what the structure is, you can always go back to previous chapters and remember there's a carbon, an R group, a variable R group, and all that good stuff. So you can always check that out later if you need to. In the genetic code, this is where it gets interesting. There are 64 messenger RNA codons. So using those 4 nucleotides of adenine, uracil, guanine, and cytosine in combinations of 3, you can have up to 64 different combinations or triplets. However, there are only 20 kinds of amino acids grand total. So what that means is even if you took the 20 amino acids and you lined them up with at least 20 messenger RNA codons, that means there are 44 other codons that must code for something. And the question is, what? And they end up being duplicates a lot of times. So we're going to take a look at an amino acid chart here in a minute, and you'll see that there are several codons that code for the exact same amino acid. And that's going to be one of your questions today on the discussion board is, why do you think there are so many different codons when there's only 20 different kinds of amino acids? So something to think about. That's your kind of your critical thinking question today. So some codons signal for the start or the end of a gene. There is actually one start codon called methionine, or MET for short. They abbreviate all these amino acids down to 3 letters. This one starts every single gene, or for the polypeptide chain. So without AUG, you won't construct a polypeptide chain. You won't get anything to assemble without this one. So it's very, very important. Then you have 3 stop codons. You have UAA, UAG, and UGA. They code for absolutely no amino acid whatsoever. Kind of weird, yes, I know. But there are tRNAs with the anticodon. They're going to match up with this. But unfortunately, there is no anticodon that can match with these. So it's a built-in stop mechanism, just like a race. You have to have a start line and finish line. In this case, we actually have 3 possible stop lines, which is kind of cool. So more than one termination point. So again, we look at the picture. You got your promoter that tells the RNA polymerase where to bind. You've got your different exons, gene 1, 2, and 3, that you're going to be used in combination to once again make different proteins if need be. And then you've got the introns, which are the gaps in between them, the non-coding DNA. And then finally, the termination sequence. All right. So if you notice in every single gene down here below, they have a stop codon and they have a start codon. So this is the amino acid chart yet again. As you look at it, I want you to pay close attention as to you have uracil, uracil, uracil for a codon. That codes for phenylalanine, which we're going to talk about. And then you notice UUC also does that. Okay, but then you look right below it, you got leucine, which is UUA, UUG, all of which of those codons code for the exact same amino acid. So there is a design or mechanism built into this, which is kind of cool. But again, as you read this amino acid chart, if I were to give you a thing as a codon such as AUG, what you need to do is you use the first base, which was A. You start over here on the left side, and you know it's going to be in this row right here somewhere. U is the second one, so you use the second base pair in this column, so you know it's going to be in this box right here by process elimination so far. And then we're looking for the G. You go to the third base pair, which is always going to be on the right side, and it tells us it's right there. So if you forgot how to read the amino acid chart, it's pretty simple. Just use and follow the directions. Right? That may be part of an assignment that we do a little later. But again, there are only three stop codons which code for no amino acid whatsoever. Therefore, it can't put one in there. It just cuts it loose. And here's the big long list. So sometimes you'll see these, like I said, three-letter abbreviations like ALA, ARG. Those actually code for specific names. All right, so as you look closely at them, you can see that they're big, long scientific words. No, I do not expect you to memorize any of these. As you get to maybe a genetics class in college or something, you might have to know the specific names, but not here. Okay, three-letter abbreviations, or I'll give you the information whatsoever. All right. There is also different letters that can be assigned to the specific amino acids. That's for another form that we don't use, but you may again use that in genetics class in college. From DNA to RNA to amino acids. So again, we're going from the DNA. We go from transcribe DNA to messenger RNA. The messenger RNA, we're going to then translate it into our polypeptide chain. So again, reading the codons one by one and attaching those amino acids to it to make our polypeptide chain. Now, RNA and tRNA, we've been talking about those, and they really haven't come into play yet, but here's how this big picture all fits together. We know that the tRNAs deliver the amino acids to the ribosomes, and the ribosome is kind of like the factory where everything's built. So the tRNAs, they must read the codon on the messenger RNA, and they therefore have what is called the anticodon, which is the opposite. So if it was AUG, it would be UAC on the anticodon, the complete opposite. And that brings in the specific amino acid methionine. All right, so there's a built-in mechanism to get the right thing for the right protein. So ribosomes, which leak the amino acids into the polypeptide chain, so the factory, the rRNA, will actually take those amino acids and induce that chemical bonding, that reaction, to string them all together. And we're going to talk about this process here in a second of how it all comes together and works together. Again, this is the structure of tRNA, which is kind of cool. It's just a long folded weird looking thing, and again the TRP is amino acid, but again this is what a entire tRNA would look like, this weird structure. And a lot of people fail to realize that the tRNA, rRNA, and the mRNA are all transcribed from the DNA. So somewhere in your DNA you have a gene for messenger RNA, which will turn to messenger RNA. You have a gene for the tRNA. You also have a gene for the rRNA, the ribosomes. And again, if you remember we talked about ribosomes, we said they're made in the nucleolus and they're shipped out to the rough endoplasmic reticulum. And the reason why it looks rough is because it has ribosomes. And ribosomes help produce proteins. So again, it's kind of a long roundabout thing, but all of these things, these RNAs, are made with nucleotides of RNA, and they are transcribed from the DNA somewhere in there. This is just a picture kind of showing you that the tRNA and the rRNA, or ribosome, will actually pair up together and actually help read this little strand right here, which is the messenger RNA, and it will start combining those amino acids together. So if this was a real life view that you could get way down there and see it, this is what it would be. All right, so take home message. What roles do the messenger RNA, tRNA, and rRNA play during translation? So turning that messenger RNA into a polypeptide chain. The messenger RNA carries proteins building information. The bases in messenger RNA are read in sets of 3 called codons, or their triplets. The code for those amino acids to, once again, make that entire polypeptide chain. There are 20 amino acids, but there are 64 different possible codon combinations you can have. Ribosomes, which consist of 2 subunits of RNA, rRNA, and proteins, assemble amino acids into polypeptide chains. They're the factory where it all comes together. The tRNA has the anticodon complementary to a messenger RNA codon. So putting this into terms that are a little more simpler that I like to think about is that the messenger RNA is the assembly line. And assembly line, as you remember, learning about that back in the history days, is where parts are put onto it, and as you put the parts on it, it becomes more elaborate and starts becoming more of that final product that you want. One amino acid is not a protein. You need several of them to fold to make a protein. So simply just having a amino acid ain't going to work. You need to continually build upon this strand to make it a fully functional thing. So that's the messenger RNA. The factory is where the assembly line is housed, and that's the rRNA, the factory itself. But how do you get parts to the factory? Truck drivers, tRNA. tRNA brings in those amino acids. So again, you got to have parts coming in, you got to have parts assembled, but you got to have a place to do it all. That's the factory. Okay, so that's the way I like to think of it. Makes it simple for me to remember and to use and think about the mechanisms behind this. Any questions so far? All right, if you have any, just post them in the chat and I'll get to them. There's a little bit of delay and lag in the video again, so if I start going and I come back to it, that's why. Okay, look at this. Oh man, being you guys liked Professor Kitty so much last time, I thought I'd throw him on here again. But again, so why should we care about this coronavirus? Doesn't affect cats. Turns out it doesn't so far. But again, cats, they need us to change their litter box. So I guess Professor Kitty will maybe save us. Maybe, I'm not sure. But just talking a little bit about that, of why a virus may not affect all species the same. We've kind of mentioned this in class before, that some viruses can be transmitted from species to species, but not necessarily all of them. As of right now, the CDC, I looked it up, said they have no indication that dogs or cats can carry the virus, nor can it be transmitted to them. And that could be just, again, the mechanism behind of how viruses are transmitted. Some were talking about the microbial bacteria in your gut, whether we have them or cats have them or dogs don't have them, whatever, for whatever reason. That may be how some of these things could be transmitted to. But yeah, it could come down to simply proteins as well, that maybe we have proteins to block it, maybe they have proteins to block it that we just don't have. So again, each species is different and unique, and that's variability within the population. So again, a virus may not wipe out everything on Earth. There could still be cats. So there you go, just a little bit about Professor Kitty and the cats. Emily asked, do you have a cat or did you kill yours? No, I still have my cat named Turdy. We rescued him from the school. He took a truck ride in when he was a little kitten, and nobody claimed him, so we took him in, and we still have our cat named Turdy. Yes, his name is Turdy. So, good times. Thank you, Emily, for the very insightful question there. All right, we're going to move on then. I didn't see any other questions. Translation again, so RNA to protein. So the order of the codons in the messenger RNA determines the order of the amino acids in the polypeptide chain. So again, making a very specific protein as we move forward. Translation occurs in the cytoplasm of the cells. And again, that messenger RNA has that poly A tail and that guanine cap on the 5' end to ensure its integrity. It occurs in 3 stages, this whole process of translation: initiation, elongation, and termination. So we're going to talk about those. So initiation is the initiation complex is formed. So in the rRNA, there's actually two subunits, the smaller one and the bigger one. So the small ribosomal subunit binds to the messenger RNA. The anticodon of the initiator tRNA pairs with the start codon of AUG to get this whole thing going. And then the large ribosomal subunit sandwiches on top of it, so it wraps around the messenger RNA altogether. So as you can see in this picture, you got the small subunit, tRNA, bringing in methionine, and then we have some GTP. Ooh, GTP, not ATP, guanosine triphosphate. Again, I told you this is used in the body. Here you go. There's one place where it's used. And then we have the large subunit attaching to it. And then you have three sites. You have the E, the P, which is right here, and the A site. Okay, we're going to talk about those three sites here in a second. Elongation, the ribosomes assemble a polypeptide chain as it moves along. So these RNA subunits will actually start gliding across and start moving down further and further. Okay, so the initiator tRNA carries the methionine, and then the ribosome joins each amino acid that comes in next with the next tRNA to the next one. So we have the next tRNA will come right into the A site. It will shift down once it does so and bond these two together, and then the E site is like the ejection site, kicks everything out. So as you can see here, when the ribosome encounters a stop codon, a polypeptide synthesis ends. So the release factor binds to the ribosome. The enzymes detach the messenger RNA and polypeptide chain from the ribosome itself. So again, we get this polypeptide chain being made here, right? And as it slides down, the tRNA lines up in the E and gets ejected, and it just continues on. And the next one comes in, and so forth. But then the release factor comes in here because it can't find a tRNA to match up with it, and it cuts it loose. So then the tRNA is ejected. You have your free polypeptide chain. The ribosomal subunits release. The messenger RNA can then be retranslated if it wants, or degraded back into this nucleotides to synthesize a new one. But that free polypeptide chain will then be finished out. It will go from, like I said, a lot of times the rough endoplasmic reticulum to the Golgi, where it will be folded, and then once again modified and packaged into sacs called vesicles. And that could go within the cell or outside the cell, wherever it needs to go. Okay, so again, there's just showing you how it all works. Bringing in the new one as it pops in, you have the peptide bond occurring, and then you have the next one coming in, but you're releasing the other tRNA, and then you have two in there, and you're starting to build this amino acid chain. And then the next one's released, new tRNA comes in, we're starting to build that chain. And then finally, we're starting to get a long polypeptide chain, which can become a protein. So we've talked about mutations sometimes and how you can have a premature stop codon occur in a mutation. When that happens, your polypeptide chain will not be as long as it's supposed to be, which means it can either A, not fold at all, or B, fold incorrectly, and therefore limit or inhibit altogether the function of that protein. So just another picture here showing you the overall flow. You got transcription occurring in the nucleus. We have large nuclear pores in the nuclear envelope to allow that messenger RNA to escape out into the cytoplasm. Again, got some protective measures, poly A tail, guanine cap. We've got the ribosomal subunits attaching, the tRNAs bringing those amino acids. And again, we can see the polypeptide chains being made, just like we saw in the previous picture, like that Christmas tree. So it's not just one and done. It can be, a lot of times, a mini polypeptide chains being made because you have multiple uses for those proteins in a cell or body. I'm going to stop here at the video. The video does a nice job summarizing everything. If you want to give it a watch, you can. I didn't want to bore you with watching a video within a video, so you can just check out the notes and summarize all that together for translation. I think it also talks about transcription as well, but it does a pretty good job. So I'm going to open up to questions, and if you have none, I'll cut you loose. So ask away. Otherwise, I hope you guys have been still enjoying the break. There's lots going on, I know, with the coronavirus, but we've had some confirmed cases in Macon County, so just be careful out there. Wash your hands. Try not to lick doorknobs and windows. Looking at you, Emily, so. In other news, I seen that the Kansas City Chiefs signed former XFL player um, Jordan Teamu, which was the quarterback for the Battlehawks, the XFL team. So pretty excited about that. All right. Any other questions? Again, you guys can always email. If you stick around, you'll have an opportunity to take the video quiz. Again, use all your resources, use your notes, use the PowerPoint slides. It's just there to guide you to make sure you're getting the right stuff. In addition to that, there's also the discussion board. This week on the discussion board, I encourage you to try a what if question or a critical thinking question, because it was brought to my attention that you know everybody's answers are exactly the same after you make a question. Yeah, if they're just factual based, they will be about all the same, and I understand that. So try to develop a question that's a little deeper, like a what if or a critical thinking of like what's your opinion even. So that will really stimulate some conversations. Okay, well, I don't see any other questions, so hey, you guys enjoy the rest of your weekend, and we'll see you back on Tuesday at 1 o'clock again. All right? Thank you guys for tuning in. I really do appreciate it. Um, I know it's not ideal, and you guys are doing a great job, so thank you for bettering yourselves over this break. It's not easy to stay on top of things, but you're doing a great job, so keep up the good work.